All right, we're a few minutes past the hour, so I think let's get started. So today we'll have presentation from three of our staff curators at the Broad for ClinGen, who are representing the Skeletal Disorders Clinical Domain Working Group, and we'll be discussing the progress of two of the gene curation expert panels, the Skeletal Disorders panel and the Craniofacial Malformations. So that'll be presented by Julie, Vanessa, and Kezang in a moment. And then other than that, in terms of announcements, the gene curation SOP V12 is now live. So we had Marina and Christine on the call last fortnight talking about updates. So that's now all officially available, and Kezang's just put the link in the chat. And then other than that, our next meeting falls February 26th at 12 p.m. Eastern Standard Time, um, where we'll get two updates. We'll get the ClinGen Code of Conduct and what's been done in that regards, and then also talking about allele frequency cutoffs from in relation to the secondary findings genes. Okay, so that's it. I will pass it on to the staff curators whenever you're ready. And if anyone has any questions, for now, to just put it in a chat and we'll get to them at the end of the presentations.
All right. Good afternoon, everyone. My name is Vanessa, and along with Julie and Kezang, we'll be presenting the current progress of the ClinGen Skeletal Disorders Clinical Domain Working Group. All right. So the main goals of the CDWG is to create clinically relevant and actionable genes and variants, primarily affecting the cartilage and the bone. And within this working domain, there are two GCEPs, which have the task to look at genes implicated in both skeletal disorders as well as the craniofacial malformations. So those would be the craniofacial malformations GCEP and the skeletal disorders GCEP. And here we just have an overview of the chairs, coordinators, and the biocurators. So at the top, we have the co-chairs who are Doctors Deepika Virkat, Carlos Ferrer Lopez, and Karen Heath. We also have the craniofacial malformations GCEP chairs, which are Doctors Pedro Sanchez, Ethlyn Wang Jabs, and myself and Julie coordinate these expert panels. We also have 14 total biocurators, which are shown at the bottom. A majority of them are volunteers with some skeletal-related experience before they begin curating for these two GCEPs. And here I've just shown a map of where these members across both GCEPs come from. So we have 61 total members as of January of this year across 16 countries, which come from North America, Central America, and South America, as well as Europe, Africa, Middle East, and Asia. And new members are accepted upon review of their CVs, and an interest or statement of interest is passed on to the co-chairs, and they review that, and then they accept or decline the new members. And the members who are part of this two GCEPs include clinicians with experience in either the skeletal disorders or craniofacial malformations, as well as diagnostic lab experts with relevant genetic testing experience, ClinGen framework experts, as well as our volunteers who are trained in gene curation. So go on and jump into the skeletal disorders GCEP. Okay, so for this GCEP, the scope of work is to review all 437 genes that are listed in the latest version of the Nosology of Genetic Skeletal Disorders, beginning with the most common genes associated with skeletal dysplasias. So our first round was curating 9 genes, accounting for the most frequently encountered skeletal dysplasias. So these were call A1A, call A1, call 1A2, sorry, call 2A1, FGFR3, SLC26A2, TRPV4, call MP, ALPL, and SOX9. And subsequent rounds of curations will include cell disorders caused by abnormalities in the cilia and ciliary signaling, and that would include short-ribed polydactyly syndrome, short-ribed thoracic dysplasia, chondroectodermal dysplasias, as well as orofacial digital syndromes. And the GCEP will also review dysplasias with multiple joint dislocations, which include the SEMDs. And within this GCEP, there are a total of 38 total members, and 12 of them being biocurators and 26 of them being skeletal disorders expert panels across the world.
Yeah, so I'll take over from here. So I'll be talking more about our curation progress. This is a snapshot of our overall curation progress within the skeletal disorders GCEP. To date, we have 57 gene disease relationships published on the website. As mentioned by Vanessa, in round one, we focused on 9 genes, which included 26 GDRs. And currently, we are in round two, where we are considering 39 GDRs, and of note, 18 of them have a primary curation published by another GCEP. So this reflects the overlap between skeletal phenotypes and other clinical domains. The next slide. So this slide shows a more granular view of our curation progress across genes. You can see the variability in classification strengths ranging from limited and moderate to definitive. Many of the core skeletal genes reach definitive classifications, reflecting decades of clinical and molecular evidence. So the genetic scores is highlighted in black, and the experimental score is highlighted in gray. And the blue line across the chart, the scores below the blue line reflects limited classifications, and those that fall between red and the blue line are moderate GDRs, and anything at the red line or above the red line represents definitive classifications. Yeah, so getting into our curation progress, we followed the standard ClinGen gene disease validity framework with additional skeletal specific considerations. So we begin with pre-curation, where lumping and splitting decisions are made using the International Skeletal Dysplasia Society nosology and ClinGen guidelines, which are based on molecular mechanism, phenotypic variability, and inheritance patterns. And then this is followed by curation, where each gene disease relationship is evaluated by a curator using the ClinGen SOP. And this also incorporates the skeletal disorders scoring adjustments. Next, these gene curations are presented to the experts for expert review and consensus on our monthly GCEP calls. And finally, these approved curations and evidence summaries are published to the ClinGen website. And these curations undergo periodic recuration to reflect any new data. Next slide. So we have published our round one results to the Journal of Bone and Mineral Research just last year. And this manuscript outlines our gene disease validity classifications of the commonly tested collagen and osteogenesis imperfecta genes, and describes several of our skeletal specific scoring considerations. So I'll be getting more into detail on the next few slides. Yeah. So this shows our skeletal GCEP specific scoring upgrades We implemented scoring refinements within the ClinGen framework to allow biologically meaningful distinctions that reflect the well-established disease mechanisms in skeletal genetics. So apart from the general de novo status and functional data upgrades in the ClinGen framework, we also awarded point upgrades for radiographic specificity and for variants in the GlyXY triplet repeat domain in collagenopathies. So the first upgrade would either get zero point 0.4 points or zero point 0.5 points, depending on the type of variant, and the subsequent upgrade would get another 0.5 points. So for radiographic specificity, the skeletal GCEP awarded point upgrades of 0.5 points after reviewing the evidence and if the radiographic evidence showed features highly consistent with the disease and features that differentiated these gene disease relationship from others in differential diagnosis. And point upgrades for collagenopathies were awarded for variants that affect the glycine residues within the triple helical domain of COL1A1, COL1A2, and COL2A1. The glycine residues within the triple helical domain of alpha genes are important for collagen polypeptide assembly into triple helices. So, and some of these collagen gene curations were at risk of moderate classifications despite the well-characterized nature of these gene disease relationships. So it was important that we upgrade the scores of these CLY-XY variants rather than counting them as a typical missense variant, which would lead in lower cumulative scores. So next slide. So this slide summarizes the results of the 9 genes included in round 1. These genes were some of the most frequently encountered skeletal disorders in clinical practice. So using the ISDS nosology as foundation, we pre-curated these 9 genes and curated 26 gene disease relationships. 22 out of the 26, which is about 85% of the GDRs, were definitive. 2 had moderate relationships, and 2 were limited. And the GDRs without a primary skeletal phenotype was excluded from round 1, and the FGFR3 craniofacial conditions were curated separately by the Craniofacial Malformations GCEP. Moving on, so this chart illustrates the gene disease validity classifications across the curated genes in round 1. Again, most of the classic and well-studied genes reach definitive classification with just genetic evidence. If you look at the chart here, you see that the black portion of the bars reach the red line for most of the definitive gene disease relationships. And where classifications were lower, they typically reflected smaller case numbers and clinical and genetic reports from additional probands and their families are needed to upgrade these gene disease relationships to definitive. Moving on to the next slide. So one of our biggest challenges during round 1 was reconciling traditional ISDS nosology with the ClinGen gene-centered lumping and splitting framework. So the ISDS system established in 1969 has more than 500 genes and is a clinically driven, phenotype-driven system, and this maintains distinct diagnostic entities, often resulting in many different conditions per gene. This nosology is widely followed by clinicians and researchers around the world and remains critical for the diagnosis and management of clinically distinct entities. However, the skeletal disorders GCEP frequently lumped more genes in these relationships than ISTS nosology based on overlapping phenotypic variability, shared molecular mechanisms, and consistent inheritance patterns. Many members of our GCEP are also members of the nosology committee, and they largely agree that the skeletal disorders GCEP constitutes a logical evolution in the establishment of gene curations. And balancing these historical clinical classifications with molecular understanding required careful and nuanced decisions from our experts. To the next slide, yeah. So this slide highlights some examples of how we approach lumping and splitting decisions in our GCEP. So in some cases, shared biology and overlapping phenotype supported lumping into single gene disease relationships. The SLC 26A2 variants cause a spectrum of skeletal dysplasias ranging from the lethal achondrogenesis type 1B to non-lethal multiple epiphyseal dysplasia. Although these entities are different, are distinct in the ISTS nosology, they share overlapping phenotype, radiographic features, molecular mechanisms, and inheritance pattern. So we lumped these two into a single GDR as autosomal recessive SLC26A2 related skeletal dysplasia. Similarly, comp-related pseudo-achondroplasia and comp-related multiple epiphyseal dysplasia were also lumped due to phenotypic overlap and lack of clear locus-specific genotype phenotype correlations, and was curated under the single term comp-related skeletal dysplasia. In other examples, distinct mechanisms or inheritance patterns justified splitting. So as you can see, for SOX9, loss of function coding variants are common in campomylic dysplasia, while non-coding deletions that reduce expression are seen in pyroban sequence, while non-coding regulatory variants that drive nearby gene expression have been reported in Cook's syndrome. So these three were split out. And ALPL-related hypophosphatasia was split based on the mode of inheritance. So the ALPL-related, the autosomal dominant form of the disease involves mild to moderate severity and is usually caused by a dominant negative or haploinsufficiency. Whereas the recessive form was caused by loss of function variants and involves perinatal lethal or moderate to severe hypophosphatasia. And for collagen genes, these provide another example where structured lumping and splitting was applied based on mechanism and radiographic pattern. So COL1A1 and COL1A2 related osteogenesis imperfecta types 1, 2, 3, and 4 were lumped under a single GDR. Whereas the classic type Ehlers-Danlos syndrome, articulatia type Ehlers-Danlos syndrome, and combined OI and EDS were all lumped as either COL1A1 or COL1A2 related EDS. And there were other disease assertions that were split out, the COL1A1 related cafe disease, and COL1A2 related EDS, the cardiac valvular type, was split out to two distinct inheritance pattern or molecular mechanisms. And finally, COL2A1 had the most split GDRs of any gene reviewed in this round of curation, based on distinct clinical features and molecular mechanisms, which we'll get into in the next few slides. And I'll hand it off to Vanessa at this point.
Okay. So I'll talk about the COL2A1 gene curation, which Kezang mentioned had the most distinct curations. So this is a collagen type 2 alpha 1, and it codes for one of the primary collagens in the cartilage. And in OMM, as you can see here, there are 15 total phenotypes that result from mutation in this gene. And they can range from mild joint diseases to lethal skeletal dysplasias. So here we can see this table, which shows the different curated GDRs, and the ones that are highlighted in green were lumped. So out of the 15, we had 8 total distinct GDRs that were curated, and you can see that they have differences in molecular mechanism, and for the ones that are split out, they have very distinct clinical presentation as well as management. So for example, here for the spondylometaphyseal dysplasias, for these two, they have the missense variants in the glycine XY repeat motif, which causes substitution in glycine to a bulkier amino acids, interfering with the triple helix formation. And that is a dominant negative mechanism. Same thing with the other spondylometaphyseal dysplasia here, caused by a single missense variant in the Y position of the glycine XY repeat motif. And for the other GDRs that are not highlighted, they have very distinct clinical presentation, as well as the molecular mechanisms are different, although some of them have an overlap. For example, for the plantospondylic dysplasia Tshering type has the same mechanism as the other spondylometaphyseal dysplasias. However, it has a different presentation, so therefore it was split out. And the others have differences in molecular mechanisms. And so here, this is what it looks like on the ClinGen website. So although they have the same mode of inheritance, they did have differences that would lead them to be split out into multiple distinct GDRs. And for 7 of these curated entities, they met a definitive classification, and one of them met a moderate classification, which is due for recuration. So this GCEP has also looked into the skeletal ciliopathies, and ciliopathies in general are a series of disorders that are characterized by variants in gene, coding components of the primary cilium. And skeletal ciliopathies are just a subtype of ciliopathies, and they mainly affect the skeleton, with common radiographical findings such as short ribs, short limbs, and short digits with or without polydactyly. And out of the 39 ciliopathy genes that are listed in the current revision of the nosology, there are 39 total genes. And 76% of them have been curated by ClinGen, and 90% of them, or 20 of the 22 that have been curated, have met a definitive final classification, with a few of them meeting moderate and limited. And as we're creating these GDRs, we have some challenges that we are encountering, which include significant overlap of gene interest across multiple ClinGen GCEPs. So this could be the kidney cystic encephalopathies GCEP or retina, as well as mortal ciliopathy GCEP, sorry. There's also challenges in disease naming, as well as lumping and splitting. So those are the challenges that we're encountering as we curate these GDRs. And here we have a few genes that have been created by a ClinGen GCEP, the skeletal GCEP. And we can see here as the other figures, the black would be the genetic evidence and the gray would be the experimental evidence, with the 12 point here on the y-axis being the definitive classification being met and blue being limited. So for example, here for the IFT81 gene, it had a total of about 7 points, maybe 6 point something points of genetic evidence, and the rest was experimental evidence, and that led to a final total points of 8, which is around limited or moderate, if I'm not wrong. So that is just an example of how we have curated the skeletal ciliopathies. And as for the conclusions and future directions, so we have completed the creation of formally tested collagen and osteogenesis imperfecta genes and published them to the Journal of Bone and Mineral Research last year. Looking into the future, we'd like to complete the creation of skeletal disorders caused by abnormalities of the cilia and ciliary signaling with engagement of the ISDS, OMIM, as well as MONDO, to establish consistent in disease naming conventions, as well as terminologies for ciliopathies and skeletal dysplasias. We'd also want to curate genes implicated in dysplasias with multiple joint dislocations in the future, and also begin recreating GDRs with moderate or below classifications and older than 3 years. And this year we'll be presenting our outcomes to the 2026 ASBMR annual meeting, which will be held in Boston. So that would be all, and I'll pass it on to Julie to talk about the craniofacial malformations GCEP.
Yep. So I'll be talking about the craniofacial malformations group, similar components as Vanessa and Kezang talked about the skeletal group for. So just for an introduction, the craniofacial malformations GCEP, commonly referred to as the Cranio GCEP, was formed in 2020 and evaluates genes associated with craniofacial malformations, as you would anticipate. Initial curations focused on genes associated with craniosynostosis and skull anomalies. Later stages of curation have focused on a broader scope of conditions causing, in addition to craniosynostosis/skull anomalies, malformations of the ear, midface, jaw, mouth, lip, and/or palate. So this is our GCEP membership. Dr. Sanchez and Dr. Jabs are our chairs. I'm our coordinator, and we have several curators as well. So for the GCEP membership, we are a truly international group with 31 members coming from 6 countries, including the US, Canada, France, China, Sweden, and England. Members include clinicians with expertise in craniofacial disorders, laboratory scientists with expertise in genetic testing and experimental studies, ClinGen framework experts, as well as staff and volunteer biocurators. So talking about our curation efforts to date, so the GCEP has curated and published 52 gene disease relationships, GDRs, on the ClinGen website, and these are GDRs from rounds 1, 2, and 3. And the group is currently curating more genes in round 3. I'll go more in depth on this in future slides. So round 1 curation focused on genes associated with craniosynostosis and skull anomalies, and it included 12 genes. And so 23 GDRs, an average of 2 relationships per gene, were curated. Most GDRs, so 17 GDRs, were classified as definitive, 3 as moderate, and 3 as limited. None were classified as disputed, refuted, or no known GDR. So 19 GDRs had an autosomal dominant inheritance pattern, 3 an autosomal recessive inheritance, and 1 an X-linked inheritance. And I included this visual below just to show the different genes that we've curated and the number of assertions for each of the genes that have met each classification, again, emphasizing that most curations have been definitive, but some moderate and limited. So round 2 curation focused on genes associated with frontonasal dysplasia/dysostosis and craniosynostosis and included 12 genes. And so 15 GDRs, an average of 1 relationship per gene, were curated for. So a lot less GDRs curated per gene than in round 1. And similarly, the majority, 12 GDRs, were classified as definitive, 2 as moderate, and 1 as disputed. No GDRs were classified as limited, refuted, or no known GDR. And in terms of the inheritance pattern, there was about a 50-50 split in terms of those that had an autosomal recessive inheritance versus those that had an autosomal dominant inheritance pattern. And I included the same type of visual in terms of the classifications of each gene and the number of disease assertions. So round 3 curation has focused on genes associated with craniosynostosis, dental anomalies, auricular anomalies, and hypertelorism. There are 35 genes currently on the round 3 list, so quite a bit larger in terms of number of genes than rounds 1 or 2, but the list may be modified in the future. And a lot of those genes were suggested by our experts. And 16 genes in round 3 have been curated and published thus far. And so similarly to what was shown for the skeletal disorders group, this image is a graph of the classifications of many of the gene-disease relationships curated by this group. So you can see that the black part of the bars represent genetic evidence, the gray bars represent experimental evidence. You can see the point scale, so the red dashed line represents the point threshold for definitive. Most of the genes cross that, and then the blue dashed line represents the point threshold for moderate. And some genes fall within the moderate category, some below. So I just wanted to highlight an interesting gene with many GDRs curated. So this is the FGFR2 gene, and the Cranio group actually curated it for 6 different assertions, all of which are autosomal dominant. So they're Apert syndrome, Cruzen syndrome, Pfeiffer syndrome, Beer-Stevenson cutis gyrata syndrome, bent bone dysplasia syndrome, and LAD syndrome. And so all of them were classified as definitive with the exception of LAD syndrome, which was classified as limited. So in terms of the disease assertion, in terms of the like specific phenotypic features, the mechanism of each assertion, so I included it here. So this is the first 3 assertions, the Apert, Cruzen, and Pfeiffer syndrome. So they all have a gain of function mechanism. They're all associated with craniosynostosis, but the specific like facial, digit, and other features do vary a little bit between these conditions, but just to highlight them here. And then interestingly, these are the other 3 assertions, the Beer-Stevenson cutis gyrata, bent bone dysplasia, as well as the LAD syndrome. And so the Beer-Stevenson and the bent bone dysplasia both have a gain of function mechanism, but the LAD syndrome has a dominant negative mechanism. And so these curations, all these assertions were curated separately due to the separate assertions like in the databases and literature, as well as the phenotypic differences, as well as the fact that even though almost all of them have a gain of function mechanism, the specific like way in which they cause a gain of function mechanism differs a little bit between the genes. That's why they were split off. So now to focus on the scoring upgrades specific to this group. So the Cranio group upgrades 0.5 points for radiographic evidence showing craniosynostosis or other skull phenotypes, and 0.5 points for a highly specific phenotype, and that is at the discretion of the GCEP. And the image below is just an example of a radiograph for the curation of SMO and Curry-Jones syndrome, the GCEP upgraded for, and the significant part of this radiograph is that there is a posterior fossa mass circled in red that is part of that phenotype. So now I wanted to go into a couple of interesting GDRs created by this group that were caused by one recurrent variant in the gene. So the first one is FGFR3 and Muenke syndrome, and the recurrent variant is the proline 250 arginine missense variant. This is an autosomal dominant condition, and so Muenke syndrome has notable phenotypic variability and includes types of craniosynostosis such as coronal, brachycephaly, macrocephaly, hearing loss, epilepsy, developmental/intellectual disability, inclinodactyly. And so the total score was 14 points with 8 points of genetic evidence and maxing out the experimental evidence at 6 points. And so the gene disease assertion is held up for almost 3 decades, and there are several publications of genetic evidence. Therefore, the classification of that gene was definitive. So for the next variant, or the next GDR caused by a recurrent variant, was ZSWM6 and acromyelic frontal nasal dystosis, an autosomal dominant condition. So one recurrent de novo variant in ZSWM6, arginine 1163 tryptophan, causes this condition. So acromyelic frontonasal dystosis includes features like brachycephaly, cranium bifidum, ocular nasal features, cleft lip and palate, and digit anomalies. And the outcome of this was a bit interesting. So the total score was 10.85 points with 8.85 genetic evidence points and 2 experimental ones. The gene disease assertion is held up since 2014, so a little over a decade, with 3 publications of genetic evidence as well as internal data from multiple labs. And even though it doesn't meet the point threshold for definitive, it was upgraded manually from moderate to definitive due to the phenotypic consistency, the strength of evidence, and also acknowledging the limitations in the scoring system of just having one recurrent variant, but also upgrading for the fact that it met replication over time and the number of publications. So, and I also wanted to highlight an unique and challenging gene created by this group. So the ZIC1, the zinc finger protein gene. So ZIC1 gain-of-function mutations are associated with autosomal dominant craniosynostosis with structural brain anomalies, whereas loss-of-function mutations are associated with autosomal recessive brain anomalies without craniosynostosis. And so the group created the gain-of-function disease. And it was interesting because I didn't even know about the loss-of-function disease until one of the experts had mentioned it on the call because it hadn't yet been published. And so for the gain-of-function curation, it was interesting. So in this protein is 3 exons long, and so all the mutations scored but 2 were de novo or mosaic. They were all located near the end of the protein, so between residues 348 and 406, and predicted escape nonsense mediated decay. There were some missense mutations scored, several nonsense, and 1 frame shift variant. And you can see the location of the mutations via the arrows and the different domains, such as the different zinc binding site and other domains. And this image nicely further denotes the different mutations like the amino acid changes and the frame shift and it variants, and it shows that, again, that they concentrate specifically in that region of about 350 to 410. So, and just to summarize the outcome of this curation, so mutations were upgraded 0.4 points if de novo or mosaic, and only if confirmed to be 0.9 points, since the mechanism is gain of function as opposed to loss of function, if the experimental evidence showed that the variant impacts protein function. And so for the curation outcome, we had just over 11 points of genetic evidence and 2 experimental evidence points. So the total was just over 13 points, and the gene disease assertion has held up since 2015, so just over a decade, and so the classification came out to definitive. So, and just to talk briefly about some of our manuscripts and posters. So, the round one manuscript is like almost there. We had originally submitted it, got a few edits, incorporated those edits, and then resubmitted it. And so now we're just awaiting publication, so that should be published very soon. And then posters about the Cranio GCEP scope and progress were presented at the 2022 and 2023 Creating the Clinical Genome conferences. Finally, for conclusion and future direction, so we plan to continue curating genes in round 3, focusing on the same scope that our curations that we've already done in round 3 focused on, potentially adding more genes to the list. And we will begin recreations of genes that are out of date. So genes with strong, moderate, limited, disputed, or no known GDR classifications at least 2 to 3 years after the last approval date. So depending on the specific classification. And another thing we want to do is expand our membership, particularly in terms of curators, because right now we don't have a ton of curators. But one exciting thing we've done that's new that this year is the first year for. So we've been mentoring 3 genetic counseling students in gene curation, and so they're all each going to be curating one gene for the group. So that's been really helpful. And we've started outlining the round 2 manuscript, and we soon plan to begin actually drafting it. So we've got some exciting future directions, and so that's all that I have.
Okay, thank you, Julie. Thank you, Kezang, and thank you, Vanessa. I'll open it up for questions. Does anyone on the call have any questions for the 3 of you? It's very interesting to always like see what each expert panel is working on and how unique and nuanced they can be. And I think the skeletal and craniofacial groups are no exception, where they have the challenges with lumping and splitting, disease naming, the fact that you upgrade for radiographs. I think that's something that's quite distinctive that other expert panels don't do. Or even with the collagenopathies, the use of increasing for that try repeat. Again, I think is something that's quite distinctive and interesting. Well, I can ask the question. I think I'll start off with skeletal first. So Kezang and Vanessa, you both mentioned how the lumping and splitting decisions were done based on ClinGen's rules, right? When you take into consideration the disease assertions, the phenotype variability, the mode of inheritance, and the molecular mechanism. And so that led to, if I'm correct, more lumping than splitting. Whereas the skeletal nosology, which has existed for a long time now, is very split based and very clinical focused. And you mentioned how many members in the GCEP are also a part of this nosology. And so I guess from your experience with those conversations, how that transpired, how they like, if they did end up embracing ClinGen's lumping of a lot of these gene disease relationships, and if there's plans in the future to actually have that reflected on their end. It's not just on the ClinGen side, but on the nosology side to see more lumping of some of these disease entities. Sorry.
Oh, yeah, Vanessa, please go ahead. Oh, oh yeah, as I mentioned, many of our experts are on our expert panel are on the nosology committee as well, and they largely agree that the ClinGen framework is a more reliable way of uh, uh, basically dividing up these conditions. And I believe they are discussed. There are future plans on discussing or bringing this back to the ISDS nosology. I believe on their next meeting, which Vanessa, I don't know if you have anything else to add to that.
Yeah, I don't have much to add to that. I would say also they do have their own focus on what they do with the IS DP, I believe, or their society. So we do try to meet in the middle with how we look at these, you know, conditions. So it's not just a ClinGen focused approach, it's also their own um, goals being reflected on how these conditions are being looked at in the next revision of the methodology. So the latest one was in 2023, and I think they're working on the other version in the coming years.
Right. And this is one for Cranio. So I think for the skeletal perspective, we got a sense of the challenges faced with lumping and splitting and the disease naming. Um, so Julie, from a Cranio perspective, I know you showed those recurrent hotspot variants and how if you don't, it's very easy for something to be penalized if there's just one variant, right? Even though it's like well characterized and it's very well known in the field to be a definitive disorder. Um, Barring that, is there any other like challenges or current challenges that the Cranio GCEP is facing or has faced that you can think of?
Um, I would say that there's sometimes like I feel like the gene list we have now, round three, is really good, but it's like really large. And so sometimes it's a little bit difficult to know like which gene I should schedule next or like um and also we've encountered like a few genes where we've like done like a pre-curation but then decided not to curate it because it's not like super relevant to our group. So I think just like further like curating our like list of genes to kind of narrow it down a bit and make it a little bit more manageable would be useful.
Yeah, I think that's something a few GCEPs face further on as they enter like round one, round two, round three. Like the round one tends to be the heavy hitters, the like clear ones that you definitely should be looking at. And so it makes sense as a group because it's now round three with Cranio, that it's maybe starting to get to the point where the genes aren't as clearly defined. They're probably still important to look at, but as you said, you've curated some and then decided actually it's not really appropriate for us to be looking at, and another group needs to look at it. So I think that makes sense. Okay, all right. Well, I think that's it then. Thank you again for presenting, especially on relatively short notice. I think it's always, as I said, always informative to see how other GCEPs are doing and in their clinical domain. So I'll let everyone free to enjoy the rest of their day and see you in our next call. Bye, everyone.